Good morning, everyone, and welcome to today's webinar on pharmaceutical secondary standards and how to create a COA built for strong lab foundations. My name is Megan Parrish, and I'm the senior editor for Pharma Manufacturing Magazine, which is producing today's event. Today's event is also sponsored by MilliporeSigma. Today, we're going to talk about a novel alternative to managing working standards for analytical use, which saves time and increases flexibility while maintaining a concrete traceability chain. As many of you know, pharmaceutical secondary standards are certified reference materials, or CRMs, that are expansively characterized and traceable as available to primary pharmacopoeia standards. The Pharma Secondary Standard product line includes thousands of APIs and API impurity CRMs. So, documented traceability and certification is critical for any reference standard used in critical applications. As such, pharmaceutical secondary standards are supported by a comprehensive Certificate of Analysis, or COA, which includes the mass balance, applicable traceable assays, and characterization information. The detailed COA allows flexibility for the user to choose either the certified or traceable assay value based on the specific application. To complement today's discussion, we will have a live audience-driven Q&A session at the end of the presentation, so please submit questions as they occur to you. Now, before I introduce our speaker, there are a few housekeeping issues we need to review. On the interface we're using for this webinar, you'll see several different boxes or sections. The upper left or audio section allows you to adjust the volume. Just hover over this section until you see the audio volume control to adjust it. Below that is the Ask a Question box. Anytime during today's presentation, you can type your question for our presenter right here, and we'll get to your question during the Q&A. And if you're having technical problems, you can also simply type in your question or concern in the Q&A box, and one of our technical engineers will help. Underneath the Ask a Question box is our Handouts area. Here you'll find additional information available for download. In the largest section, you'll see today's presentation slides, and below the slides, you'll see a live chat box. This is where you can chat with other webinar attendees, but please do not put questions for the Q&A session in this section because this isn't where we'll look for those. Also, you can always enlarge a section by simply clicking on the square box in the top right corner of the section next to the X. And if you accidentally close out a section, don't panic. You can get back to it by clicking the refresh button at the bottom of the screen. It's located to the left of the icons. This webcast will be archived. We encourage you to direct coworkers to the recorded presentation. It'll be available on the Pharma Manufacturing website. We've also recently added a Q&A feature to our on-demand presentation so that listeners can ask the speaker a question after the webinar, and it will be passed along to them. And one final note, we ask you to please respond to the feedback form at the end of this presentation. Your answers help us craft future events. Now I'd like to introduce today's speaker. Jenna Milliken is an R&D manager at MilliporeSigma, where she leads a team in the development of new products and first batch quality control testing of both meat and solution certified reference materials. Jenna holds a master's in chemistry and has now worked at MilliporeSigma since 2015. And with that, I will hand the floor over to Jenna.
Hello, and thank you for that introduction, Megan. Thank you, everyone, for joining in today. Today, I will be talking about pharmaceutical secondary standards, a COA built for strong lab foundations. Today, I am representing the company MilliporeSigma. I'd like to further explain that the life science business of Merck KGaA in Darmstadt, Germany, operates as MilliporeSigma in the U.S. and Canada. This is the same company that is known as Merck KGaA throughout the rest of the world. So here is our agenda for today. During today's webinar, I will be covering the following topics. First off, we will discuss the hierarchy of certified reference materials. Then we will move along to the traceability of secondary standards as working standards. Next, we will discuss pharmaceutical standard certificates of analysis and their characterization. And then finally, I have just a short bit on impurity portfolios and complex mixture CRMs. So we will get started now with the hierarchy of certified reference materials. Reference materials are available so that qualitative and quantitative testing can be performed. A reference material is available to set the standard or make a definition so that information of an unknown sample or specimen can be determined. Qualitative tests confirm identification and can be used for screening purposes. We know that an apple is an apple or an orange is an orange based on physical properties, although the picture shown here might throw you for a loop. Quantitative tests assign the amount of material present or the potency of the material. Quantitative testing may also determine the amount of a hazardous impurity present in something like a limit test. For example, in the pharmaceutical world, it is important to know exactly how much of a harmful analyte might be present, say a carcinogen, because that final material is consumed by a person. So what does testing mean? Why are reference materials necessary? Well, testing ensures safety, integrity, strength, purity, and quality. And remember, your results are only as accurate as your reference. The International Organization for Standardization, or ISO, has set guidelines for materials and harmonizes definitions between classifications of standards. So this is a diagram that I'm going to show that has the hierarchy of standards and how they are defined in relation to each other. As we move up the pyramid, the level of certification and traceability increases. So at the very bottom, we have reagent grade or research grade chemicals, which technically are not standards. These materials may or may not come with a certificate of analysis. Above the reagent grade chemicals are analytical standards, which may come with a certificate of analysis. Also, these materials are held accountable to ISO 9001, and this is a guideline that requires implementation of the quality management system, or QMS. Above that are reference materials and then certified reference materials. Both are accountable to ISO 17034, and both are manufactured by an accredited reference material provider. The main difference between these two types of standards are the certification process requirements. Both certified reference materials and reference materials consider content, stability, and homogeneity. However, certified reference materials additionally report uncertainty and traceability as well. Certified reference materials are considered to provide the highest level of accuracy, uncertainty, and traceability to an SI unit of measurement. And then finally, at the very top of the pyramid, we have primary or compendial standards, and these are standards that are issued by authorized bodies such as NMIs or pharmacopoeias, and they are ultimately traceable to the SI. Standards at this top level are authorized standards. So metrological traceability is an important concept in the world of reference materials. The International System of Units, or SI, defines the 7 units of measure as the basic set from which all other SI units can be derived. The 2 most common SI units of measure for traceability in the reference materials world are the kilogram and the mole. Metrological traceability means that measurements can be meaningfully compared across different places, at different times, by different people, and using different equipment, because we set a standard of ground zero where to measure from. The measurement result is related back to a reference through a documented and unbroken chain of calibrations tracing back to the SI unit of measurement. So there are 2 types of primary standards. So the first, a primary standard, is one that is released by an SRM or NMI in metrology, and it is a standard that is designated or widely acknowledged as having the highest metrological qualities and whose value is accepted without reference to other standards of the same quantity within a specified context. These certificates provide a property value, an associated uncertainty, and they also show the metrological traceability. In a pharmaceutical setting, a primary or compendial standard is, again, one having the highest metrological qualities whose value is accepted without reference or comparison to another standard. Compendial standards are also produced and supplied by official scientific organizations such as the EDQM in Europe, the US Pharmacopoeia, and the British Pharmacopoeia. These certificates, the compendial primary standards, routinely provide a content on their certificates. The validity of a primary standard will never be questioned, as primary standards are the authority and their value is accepted without reference. Also, these standards are immediately recognized and accepted by auditors, so they can be very handy in this sense. So primary standards can even be used to qualify other standards, is what we will get to in a bit. Compendial standards are designed to be used with monographs, and there is assurance in this, as the standard then performs exactly as expected, since they were designed according to the monograph. The compendia specifies how to use the standards and in what application, so they work, without fail. Also, many controlled labs explicitly require the use of standards that are manufactured by an authorized body. So traceability against the primary standard may be expected in a situation like this. So it's important to understand what the requirements of your given lab is. All right, and so now I'd like to come back to the title of this presentation and discuss secondary standards. So secondary standards are certified reference materials, and they check all of the boxes in being that they are evaluated for homogeneity, stability, and content, and also an associated uncertainty is determined and provided on the C of A. But what makes the secondary standards unique is that they are traceable as available to a compendial standard and additionally to the SI. So when I say traceable, I mean that the secondary standard has been directly compared against the primary compendial standard. So essentially, you could say that a secondary standard is just 1 degree removed from a primary standard, hence the name secondary. Traceable standards are for use in an analytical lab application as specified in the compendia, and use can include a variety of method verification practices, including instrument qualification and calibration, analytical method validation, system suitability testing, assays, and control checks. To show this thought a little differently, we can refer to this flowchart. First off, we have the primary compendial standard at the very top. From these primary standards, company in-house working standards and working standard solutions can be qualified. These two buckets include the pharma secondary standards and the CRM mixtures that include active pharmaceutical ingredients and related compounds. Any of these three standards, the primary standard or the CRM pharma secondary standard or mixture, can be used in regulated labs for routine use. The use of secondary or working standards in the place of primary standards is a valid option that has been recognized by regulatory bodies. In the Food and Drug Administration Guide for Industry, it is stated that, quote, a reference standard, i.e. a primary standard, may be obtained from the USPNF or other sources. A working standard, in-house or secondary standard, is a standard that is qualified against and used instead of the reference standard. Further support from the USP in General Chapter 1010 that states, quote, users apply a range of strategies and practices to assure articles achieve and maintain conformance with compendial requirements. Strategies and practices can include the use of a secondary standard traceable to the USP reference standard to supplement or support any testing undertaken for the purpose of conclusively demonstrating conformance to applicable compendial standards. And then my final example is from the EDQM, Chapter 512, which defines a secondary standard as a standard established by comparison with a primary standard. A secondary standard may be used for routine quality control purposes for any of the uses described, provided that it is established with reference to the primary standard. These are just 3 examples of support for the use of a secondary standard, and it shows that a secondary standard is valid. So to summarize the hierarchy of reference materials, we can again consider the pyramid of standards. As we move up the pyramid, the more parameters are evaluated, the traceability more thoroughly defined and evaluated. At the top, we have compendial standards and NMI standards, and then certified reference materials are materials that are fully evaluated for, and you can see in the checkboxes here, purity, identity, content, stability, homogeneity, uncertainty, and traceability to both the SI or the primary standard in the case of a secondary standard. Please remember, your result is only as accurate as your standard, so it's important to choose a standard that has been thoroughly evaluated and is traceable. The next topic we will discuss is the traceability of secondary standards as working standards, and we will take a closer look at various values on the certificate of analysis. So secondary pharmaceutical standards come with a very detailed C of A. Here is a picture of the first and second pages of the C of A packet. A's usually range between 5 and 7 pages. These pages describe all analytical parameters used in the certified mass balance value and the assay value. This is a couple pages of the C of A for the secondary standard chloroquine phosphate, a drug that we have been hearing a lot about lately. So there are two types of value assignments on the C of A, the certified mass balance and the traceable assay. The certified value is the mass balance, which is traceable through the SI, like we previously discussed. So this value includes the expanded uncertainty associated with the assignment of purity. In the case of this particular lot of secondary standard chloroquine phosphate, the certified purity is 99.7 plus or minus 0.1%. The next value, the traceability assay, shows the comparative assignment to the current compendial lot. This material was assayed against the USP chloroquine phosphate and against lot number R075S0. So that's the current lot of the USP. When directly compared to the primary standard, secondary standard of chloroquine phosphate is assigned to have a value of 99.6%. Notice that there is no expanded uncertainty associated with this value, and that is because the traceable assay is not the certified value on the pharmaceutical secondary standards, C of A. Here we have a secondary standard amlodipine-related compound C, which is also known as amlodipine impurity G. Often the case that a particular analyte will have maybe one name has been defined by the USP and another name has been defined by the EP. So the certified purity for PHR 2052 assigned by mass balance and is traceable to the SI unit, in this case the kilogram, as the assignment has come from the mass balance using a gravimetric approach. Second form of traceability is by comparison to the current compendial lots. In this case, PHR 2052 corresponds to the USP amlodipine-related compound C and the EP amlodipine impurity G. The USP declares a content on their primary standard as 0.99 mg per mg. So the secondary standard PHR 2052 is assigned at 99.4% when directly compared to both the USP and EP lots. You may notice an asterisk next to the EP content. In the case of the EP standard, no content was declared on the certificate of analysis for the EP product. As a result, the assigned content of the EP certified reference standard was determined by assay against the USP standard. Next, I would like to walk through secondary pharmaceutical standards certificate of analysis and discuss the characterization process in depth. During the certification process, compendial standards are tested against the monograph it is identified with. For example, when a product such as acetaminophen is tested, the procedures in that acetaminophen monograph are followed. If it's the case that no compendial monograph is available, then methods may be developed from the API monograph and validated. So the procedures typically include identification and purity or potency testing. So typical identity testing includes NMR, IR, mass spec, or other techniques such as optical rotation, melting point, boiling point, density, and refractive index. These are the qualitative tests. Then there might be multiple methods associated with the purity or potency testing in a given monograph. So a mass balance technique would include chromatographic purity and residual measurements. Compendial purity methods also include titrations. However, a titration method could be less selective than a chromatographic method. So If it's the case where a chromatographic method is available, it's generally preferred, and that is the one that is considered. Also, chromatography methods are traceable to the compendial standard, as peak areas can be directly compared. The peak area of the assay of the secondary standard can be directly compared to the peak area of the primary standard. Certified purity is determined by the mass balance, which is calculated as the purity fraction determined by residue on ignition, loss on drying or Karl Fischer, residual solvents, and total chromatographic impurities. So if you were to consider mass balance in its most simple form, we would say that the content of the material is obtained by subtracting everything that is not that material from 100%. Take away all of the impurities from 100%. However, you can see that the TCI contribution factors in a different way than the other contributions in the mass balance equation. So ROI, water, and residual solvent factors contribute by a weight percent basis, while total chromatographic impurities are calculated by an area percent basis. TCI is calculated this way because HPLC methods see the peak area of an impurity relative to the sample that was prepared for HPLC, and contributions from things like inorganic salts or water may not be visible in an HPLC chromatogram. So we're looking at the fraction of impurities that are visible in comparison to the rest of the peak areas on the HPLC. So this is the mass balance equation that is used for the characterization of secondary standards. Next, I'm going to go through an example of how each contribution is measured and how it factors into the final mass balance. First, we will consider residue on ignition for adrenaline hydrochloride. I'd like to say that each area circled in blue is a direct excerpt from the secondary standards COA. We have residue on ignition that was performed to determine the contribution of inorganic content in this particular PHR, 2478. So here is a picture of a few samples burning off during the initial stages of residue on ignition. So after this stage, the crucibles will be placed in a muffle furnace where all the organic content will then be burned off. After taking the mean of 3 measurements, it was determined that the residue contribution was 0.046%. Additionally, residue could have been considered by analysis through ICP or IC instrumentation. So since these were additional ways to determine residue content, I included pictures of these instruments as well. Next, we will consider water content in the adrenaline hydrochloride. So water can be determined in a couple different ways, by loss on drying, which is a gravimetric technique, or by Karl Fischer. In the case of adrenaline, Karl Fischer was used. After taking the mean of 3 measurements, it was determined that the water contribution was 0.0097%. Now we will consider residual solvent contribution in adrenaline HCl. So a GCMS headspace method was adapted from the General Chapter 467 from the USP. The parameters are listed here in this excerpt on the secondary standard COA. So all parameters, including the column, the carrier gas flow, split ratio, and temperatures programming are detailed in the COA for ultimate transparency, so the user can see exactly what method was used to look at residual solvents. In this case, it was determined that there was not a significant contribution from any residual solvent in the adrenaline standard, so the overall residual solvent contribution will be set at 0%. And our final term to consider is total chromatographic impurities. This is the HPLC method that was used to determine chromatographic impurities present in the secondary standard on adrenaline. All analytical conditions that were used are listed on the COA, so this again is a direct excerpt. So you can see exactly what mobile phase and the ratio was used, along with the flow rate, column temperature, injection volume, and detector wavelength. It can be really handy if you have this standard but you know you want to run it on HPLC. There's a method provided for you here so that you could test it on HPLC if you'd like to. So there's a chromatogram that is shown on the COA. For the TCI, we've zoomed in so that we can see impurity contributions, and then all impurity contributions are listed here. So there were 2 unspecified impurities and a total chromatographic impurity content of 0.169% is used in calculations for mass balance. Now that we have all of our terms, we can finally put it together, determine the purity assignment for adrenaline. So we just went through how the contributions ROI, LOD, water, residual solvent, and chromatographic impurities can be determined. So we can insert those impurities into the mass balance equation and determine the purity or potency value. So the number highlighted in yellow is the certified mass balance that is shown on the front of the certificate analysis. The next thing we will consider is the uncertainty, or the plus or minus value next to the purity value that we determined. The uncertainty of the CRM is the sum of squares for the following sources of uncertainty. The uncertainty of the characterization is the UCHAR. Next term, UBB, is the uncertainties related to possible between bottle variation. And the last 2 terms are the uncertainties due to long-term storage, or LTS, and the instability during transport or short-term stability is the STS uncertainty. So the uncertainty of the characterization considers the uncertainties due to the characterization of the material. So those measurements that we talked about earlier for water and TCI and ROI, each measurement has an associated uncertainty present, and we need to include that into the assignment of the mass balance. So it is in this term, the uncertainty characterization, where the method uncertainties for the mass balance terms are included. The next uncertainty contribution is from the between bottle variation. So it cannot be assumed that each unit in a manufactured batch is identical. In fact, it must be demonstrated that the first of the batch is the same as the middle and the late parts of a batch. So certified reference materials are tested for homogeneity. So in this test, multiple units in the early, middle, and late portions of a manufactured batch are measured for homogeneity. Here is an excerpt from the C of A, where you read at the bottom, you can see that the homogeneity was considered using an HPLC method, and the sample size was 50 milligrams. Also, this is in accordance with ISO Guide 35. So the between bottle calculation involves an analysis of variance, where the square root of the mean of squares within groups, or replicates, is divided by the number of replicates, which is then multiplied by the quad root of 2, which is the ideal Gaussian coverage factor, divided by the degrees of freedom within groups. And then this whole term is divided by k equals 2 for ideal populations. And finally, the last uncertainty term comes from long-term and short-term stability factors. Short-term stability is evaluated to ensure that degradation does not occur during transport, while long-term stability is evaluated to ensure that degradation does not occur during long-term storage. These are pass-fail values, and most of the time, no contribution from these uncertainties is included. The expanded uncertainty is equal to the uncertainty of the CRM multiplied by the coverage factor, K, which is determined from the actual distribution's mean, standard deviation, and theoretical degrees of freedom, which are calculated. If the population is not behaving ideally, then this K value will increase from 2 to reflect additional uncertainties of the population. And when it does this, it creates a larger associated uncertainty with the standard at hand. So the K actual will be calculated so that it assumes a confidence interval of 95% or 2 standard deviations, so that we have an appropriate coverage factor. The plus or minus value will include 95% confidence. So the expanded uncertainty is the value that is noted on the certificate of analysis as the plus or minus value. So we've just covered how secondary standards are traceable to the mass balance to the SI kilogram, and now I'd like to discuss how secondary standards are traceable to the pharmacopoeia, back to that compendial standard. So secondary pharma standards are traceable to the compendial monograph. I have ibuprofen excerpt monographs from both the USP and EP, just the first little bit. Also, secondary pharma standards are traceable to the USP, EP, and BP primary reference standards. The secondary standards are traceable to compendial standards in multiple ways. First off, the methods on the C of A reference the compendial monograph. Analytical conditions called out in the monograph are used. So at the bottom, here is a representative niacinamide chromatogram that was ran using conditions specified in the monograph. So you can see the mobile phase uses sodium heptane sulfonic acid, which was called out in the monograph, and all other parameters were called out in the monograph. Additionally, content is assigned through comparative assay to the compendial standards. In this case, content has been assigned against the current lots of the USP, EP, and BP standards. When a new lot is released by the pharmacopoeia, the secondary standards are recertified against the new primary standard, and the updated C of A is available online. So if the USP issues a new lot of niacinamide, then the secondary standard will be requalified against that new lot, and the updated certificate will be available to view. So when monograph methods are available, pharma secondary standards are qualified using the compendial monograph method and held to the requirements of that method. So this is a chromatogram of the system suitability injection for PHR 1258 chloroquine phosphate. Per USP monograph, the system suitability was required to have all of these components present, so phenol, RCG, RCD, hydroxychloroquine sulfate, RCA, the API, and related compound E. Also, the system suitability was required to meet certain requirements, such as resolution, tailing, and standard deviation, to show that the method was behaving correctly on our chromatographic system. So upon qualification of the pharmaceutical secondary standard, PHR 1258, all of these system suitability requirements were evaluated and met. Other types of assays can be performed. For example, content can be assigned by titration. Here is an excerpt from the secondary standard C of A for sodium acetate trihydrate. The material was titrated using perchloric acid, and uncertainty has been assigned. In the case of this particular product, there are two certified values, one by titration and one by mass balance. The great thing here is that the user can pick which value is appropriate for their application. For example, if they are using a secondary standard in a titration application, then the certified value by titration may be more appropriate than the certified mass balance value, although they'll probably be very close to each other. Also, content can be assigned by UV or UV-Vis. In some monographs, content is assigned using a molar absorptivity coefficient. However, in this case, the content was assigned by direct comparison against the primary standard. USP monograph was followed, and the PHR 1196 was tested at 375 nanometers against the current compendial lots, which in this case include the USP and EP primary standards. Further traceability to the primary standards includes IR comparison for identification. So here, the secondary standard is compared to the current BP, USP, and EP batches. The spectra is stacked on the C of A so that comparison can easily be made just visually. Sometimes it is the case where compendial standards do not have a monograph available. For example, related compounds and impurities will most likely not have a dedicated compendial monograph. It is the case for flunixin related compound B that there is no monograph for the assay of the standard. And that makes sense because it's just a related compound. But in cases like these, new methods must be developed for the assay and the chromatographic purity assignment of the related compound that we're looking at. So sometimes it is the case that the API monograph could be used for development. but sometimes newly developed methods are required because of the properties of the analyte on hand. In this case, a new method for Premixed and RCB was needed to be considered because the API compendial monograph for Premixed was not particularly appropriate for the compound because you can see that the related compound B is much smaller and much more polar than the API. So the related compound B is circled in blue, and the PI structure is the last eluting peak, right next to that last eluting peak. A new method was developed and validated. In this case, a different type of selectivity was employed. It is a F5 column, which is much more suitable for a small polar molecule, and so we use that instead of the C18, which was used in the assay of the API. But in this new method, we demonstrated resolution against the API and other related compounds, related compound A considered, or C, high-range linearity for assay assignment, and low-range linearity for impurity assignments were evaluated. Method validation of new method would check for appropriate selectivity, peak shape, linearity, RSD, and other factors. Also, it can be the case where no compendial monograph, even in a pending form, or compendial standards are available. So this is the case for recently released sulfonified mesylate, which is a Parkinson's drug. So a new method was developed and validated to show selectivity between the mesylate and 3 of its related compounds listed here. As always, the method information is printed on the C of A so the end user can see what method was used to determine the total chromatographic impurities. In the case of sulfonamide mesylate, there was no compendial standard available. The only value on the certificate of analysis is just the certified mass balance, as there's not a compendial primary standard available for that traceable assay comparison. All right, and now we will move on to our fourth and final short topic, which is impurity portfolios and complex mixture CRMs. Multiple impurities can be associated with each API. For example, here is a PHR product list for impurity and related compounds associated with ibuprofen. This is, of course, just a partial list of many compounds that have been identified as potential impurities for ibuprofen. Impurities can be identified by, monographs listed out in the monographs, or by industry or other means as well. So this list I'm showing here is about 20 analytes that includes compendial and non-compendial standards. The secondary standard ibuprofen is traceable to the USP, EP, and BP standards. So we've got 3 X's showing the traceability over here. And another example would be ibuprofen impurity B. This pharma standard, PHR 1934, is traceable to the EP standard for impurity B. Sometimes it can be very challenging to find a compendial standard for one of these impurities because as we move down the list and we look at ibuprofen impurity G, you can see that even though this standard has been identified by a pharmacopoeia, there is not a compendial standard available. So even though there is no compendial standard available, a secondary standard is available, and acting as a CRM, the certified mass balance for the standard, can be used for quantitation. So this is great because you can look at ibuprofen impurity G in your materials and be able to assign a content to what amount of impurity could be present. And as you move further down the list, you will find impurities that do not have a common name. So these are ibuprofen impurities that have been identified as impurities outside of the pharmacopoeial setting. These are known as ibuprofen impurities that have been identified by industry. So the pharma secondary standards act in a truly complementary manner to primary standards and compendial standards, in that the secondary portfolios can offer impurity standards that may not be available otherwise. Next, I would like to briefly touch on some mixtures that are already prepared in solution. So in addition to the neat secondary standards, some APIs and their related compounds are available as a premixed solution standard. Premixed solutions can save an extensive amount of time and sample prep as these standards are ready to go. There's no weighing or diluting or making sure that it's in solution. These are called snap and shoot standards, and they're really convenient. So this standard that I'm showing here includes acetaminophen and 7 of its related compounds or impurities. And these impurities are certified by mass balance and traceable to the USP and EP by their original assay, so there's traceability in these working solutions as well. Components are premixed in solution for daily working standard use. And finally, we will conclude. So to wrap up here, I'd like to quickly summarize. Secondary standards are certified reference materials. These reference materials are unique in that traceability is offered to both the current primary standard and to the SI unit through mass balance. The detailed C of A is updated to the current lot of the primary standard, and the C of A offers enough detail so that the user can understand how it was qualified and in which impurities are present. So the portfolio is increasing, the secondary standard portfolio, and currently it has about 1,500 products. These catalog items include both compendial and non-compendial impurities. So thank you again for your attention. Feel free to ask me any questions that you might have. Contact information is listed on the slide along with Michael Hurst and Marcus Obkerker.
Okay, great, Jenna. Thank you so much for that thorough presentation. Now we're going to go ahead and move on to the Q&A section. Just a reminder, everyone, if you have a question, please go ahead and ask your question or type your question in the Ask a Question box on the left-hand side of the screen, and we will do our best to get to all the questions in the time we have left. Okay, Jenna, here's your first question. Which purity value is recommended for my use, the mass balance or traceable assay?
So the question of mass balance versus traceability assay value, I would say we would always recommend to use that certified mass balance value, and that is because that is the certified value on the C of A. The user ultimately has the flexibility to pick which value is most appropriate for their application. So if you selected the secondary standard because of the traceability to the USP, and you need to, show that traceability to the USP standard, then you could consider using the traceable value. However, my recommendation would always be to use the certified mass balance value because that is the certified value on the C of A. But ultimately, the user does have the flexibility to choose which value they'd like to use on the C of A.
Okay, great, Jenna, thank you. The next question we have is, are certificates of analysis shipped with the secondary standard?
So the certificates of analysis are actually not shipped as a printed form with the secondary standard. Instead, they are available online, so you can just go to the sigmaaldridge.com website, find your part number, and download the certificate there. This is better because if the traceability changes, if a new lot of the primary standard is released, then we will work to recertify the value to the current compendial lot of the new primary standard. So the certificates are not printed, but they are always available online to the most current standard.
Okay, great. And someone is wondering, are long-term and short-term stability tests performed?
Yes. So we test both long-term and short-term stability. Short-term stability tests include an accelerated test where we stress a sample in a hot and humid environment, and then we compare that to an unstressed sample. Typically, there is minimal degradation or no degradation observed. And then also we look at long-term stability, and that is just looking at a recertification, or as we can test the material after it's been on the shelf for a couple years or 18 months. We will test again to make sure that the content has stayed the same.
Okay. Now I have a question. Someone is asking about a working standard for humidity and temperature conditions with the PACT secondary standard.
So I think maybe they're wondering about what the conditions are when the secondary standards are packed. So most secondary standards are manufactured in Laramie, Wyoming, and we have a very dry climate, around 20% relative humidity. However, if one of these materials is hydroscopic or unstable, we have plenty of glove boxes where we can package these materials in as well. So in an inert and humidity controlled environment.
Okay. Next question is, which pharmacopoeias do you demonstrate traceability to?
So currently we demonstrate traceability to the United States Pharmacopoeia, the British Pharmacopoeia, and the European Pharmacopoeia.
Okay, great. Quite a few more questions that have come in. Here's one wondering about, they're wondering, how do I know if the traceability assay of a secondary standard has been requalified to a primary compendial lot?
Okay, that's a good question. So it might be that you have noticed that the lot has changed on the USP, or something that's easier to do is you can sign up for certificate of analysis updates. So you can go to the Sigma Aldrich website, and there is a C of A update email sign up, so that you can sign up for monthly pharma secondary standard C of A updates, and the C of A's are updated every time a new batch is released by a primary standard. So I don't know if I can chat that link to everyone. I think I sent that. I don't know. Can everyone see that link?
I think that I can move that link into the attendee chat so that everyone can see that. I'll go ahead and copy and paste that in there.
Maybe here. Oh, yes, I see it. Perfect.
Right. And again, everyone, we do have a Q&A function on our on-demand webinar at pharmamanufacturing.com, so you can also follow up with Jenna through that link. Okay. The next question is how to compare a working standard with primary standard when using a titration method.
Okay. So a lot of times, it depends on how the monograph is written. So a monograph that requires a titration will assign content to a material in one of two ways. So the first way that is most common, and I think used almost all of the time, is by assignment of content against a titrant. And so that titrant has been standardized against, a BAM, a primary standard. So, if you use this amount of NaOH, then your content is, this % purity, because, stoichiometry and the milligram per mole equivalency conversion. So that's most of the time how content is assigned, and so there's traceability through the compendial monograph, because you're following the compendial monograph method. However, there are a few cases that I can think of where the USP content or the USP standard is actually standardized or determined, and then that is then used to determine the content of the unknown at hand, so in our case, the secondary standard. And so it's a relative assignment based off of the USP standard. But the methods are transparent in the C of A's, so most of the time it will say that, perchloric acid or sodium hydroxide was used to determine the content of the secondary standard. And then the uncertainties that are associated with that come from, uncertainties on the, regarding the standard that was used to qualify the titrant, and then also like the weights and the volumes of titrant delivered.
Okay, good. We just have a couple questions left. Someone is wondering, what is the max primary shelf life?
The primary shelf life. Shelf life can be evaluated, ongoing. Our certificates do have a typical expiration date of about 3 years at the moment. However, it depends on the analyte at hand, of course, because each analyte is different. And so based on data that we see from long-term stability, or short-term stability, this shelf life date may be adjusted. But the value on the certificate right now has an expiration date, so the user can go to the certificate of analysis and see what the expiration date is there.
Okay. It's kind of a follow-up question, it looks like, to that. How many months maximum can be a given shelf life for a secondary reference standard after standardized against primary reference standard?
So again, I would go back to that expiration date on the C of A. The certificate may be updated against a new compendial lot, and the new C of A can be referred to then.
Okay, and then this looks like it's our last question, and then we'll wrap up. Somebody's asking about a purity when determined with mass balance, if there's a difference with the HPLC method, and they're wondering what the acceptance criteria is for this difference.
Okay. So I think the user or the question might be regarding maybe the mass balance value will have a different value than the traceable value. And again, this is up to the user to decide which value they want to use for their application. Generally, the values are very well aligned within 1% agreement. However, it can be the case where maybe there's a discrepancy between the certified mass balance and the traceability assay. And so in this case, we've dug in and tried to find the root cause of this difference. However, we do not assign content to the USP primary standard, and so if our content is significantly different according to the USP, we're not going to change this value or anything. We're going to be ultimately transparent. But typically that is not the case. Typically the two values are within 1% agreement of each other.
Okay, great. Thank you so much, Jenna, for taking the time to field all those great questions. Everyone, thank you so much for submitting the questions. This is just a final reminder to the audience, so please let us know your thoughts on this presentation by answering our brief survey. It's going to appear on your screen shortly. Your answers are really appreciated, and they do help us craft future events. And just a reminder, this webcast is going to be available at pharmamanufacturing.com. You can feel free to log in to view it, or please share it with your colleagues. We also encourage you to visit pharmamanufacturing.com to gain access to even more tools and resources aimed at helping you achieve on-the-job success. And on behalf of Pharma Manufacturing Magazine and the folks at MilliporeSigma, thank you so much for your attendance this morning, and have a great day.